and thank you for joining us today. We have the pleasure of hearing from our speaker, Abel Mebrahtu today, who is a master's student in biomedical engineering under the supervision of Dr. Walter Herzog. Before beginning his master's program here at the U of C, Abel completed a Bachelor of Science in Exercise and Health Physiology in April 2024, also from the University of Calgary. During his undergraduate degree, he conducted his honors project at the HPL under the supervision of Dr. Herzog, which is the experience that sparked his interest in continuing his research with the lab group. A fun fact about Abel is that he won first place at the Innovation for Health Hackathon for developing a machine learning-based device aimed at supporting individuals with endometriosis with the broad goal of advancing women's health. And he is also a passionate FC Barcelona supporter, and he boldly declared in his form that Messi is the greatest of all time. Let's leave that argument for a different day. Today, he will be presenting his talk titled, Can a High Aerobic Capacity Preserve the Structural Integrity of the Shoulder Joint in the Presence of a High-Fat Diet?
We should make it. Lionel Messi.
Thank you,
Ain't
Abel.
that for Lionel Richie? Well, thank you everyone for attending my seminar today. And to get us going into this, the main premise of my research is to look at osteoarthritis. Many of you might know that it's a chronic and degenerative condition that goes throughout an entire person's life. And basically, it's essentially where the cartilage between the joint degrades. And this is typically seen in some joints, such as like the knee. And as you guys can see here from on the left, you can see what a normal knee looks like and what the knee looks like with OA. And clinically, individuals diagnosed with OA typically have symptoms of joint pain, stiffness, swelling, and a reduced range of motion. And this makes it quite difficult to do daily activities, and especially activities that can maybe potentially promote like a good quality of life, such as exercise. But with osteoarthritis, it is quite linked to obesity, with it being a public health crisis with over 1 billion people worldwide being affected, and as a context for the Canadian population, 30%. And with obesity, it's typically characterized by an excess of body fat, which is usually determined by a DEXA scan, which is like the gold standard, or through a body mass index. Although obesity leads to a whole array of comorbidities, one of the more common things that we're looking at in our lab is metabolic syndrome. And for people who might not know what that is, metabolic syndrome is a cluster of diseases. The main ones we look at it are like hypertension, dyslipidemia, and hyperglycemia, and some of the other few here shown on the right of the screen. And a lot of the times people have this assumption that the link between obesity and osteoarthritis is because of the excess body weight. Think about it. If you're obese, you're putting more strain on your hips and your knees. And this greater weight leads to more forces on these weight-bearing joints, leading to that degradation of these joints, leading to OA. But with people who are obese, they also develop OA and non-weight-bearing joints, such as the hands. And with this, a lot of physicians have coined the term metabolic OA, and this is because the adipose tissues from people who are obese typically secrete adipokines and cytokines, leading to this degeneration in the joints. And commonly, many people might not know this, but the most common affected joint in the upper body is the shoulder joint. And 1 in 3 people develop shoulder OA over the age of 60, which is one reason why my research is looking at this problem. But although people have looked at metabolic OA, it's becoming more of a recent trend in the research. Researchers will look at this through preclinical models because it's hard to look at adipokines and cytokines in humans. So to do this, a lot of them gave this like high-fat, high sucrose diet, like this burger and pizza you might see on the screen, and I've looked at various different tissues. And especially the two in this presentation that I want to bring up is the joints and the muscle. As previous research has shown that the knee and the shoulder joint seem to be compromised, the subchondral bone seems to degrade, and there's changes in muscle morphology. And to look at this deeper, let's look at the knee and the shoulder joints. With the knee and shoulder joint, one common area they're looking at is the cartilage. This is that smooth, lubricated layer between the joint that allows us to make these easy movements with our joints and not hear that crackling sound as we move it with people who have OA. And one individual in our lab previously, Nada, who finished her PhD, looked at these joints, specifically the shoulder joint. And she's been able to see that when we look at these joints, both in the male and the female, there's a significant amount of degeneration seen with this high-fat, high-sucrose diet in both groups, more so with the males. And here we're able to see that this preclinical model in the shoulder joint can be potentially be used to look at metabolic OA. But even though this is from a quantitative view, most people don't know what it looks like under these animals. So if I show you this picture here on the left, this is what a healthy shoulder joint looks like. We see that nice cartilage here shown on the red, follow this red dot I have, where we're able to see this like red pinkish cartilage. And here below is this bone. Now here on the right is what OA degeneration looks like in the shoulder joints. Here we have this collapsed cartilage dotted in blue. And at the below of this is the subchondral bone cyst. These are things that we would not want to see in someone living who's healthy, and this kind of has a compromisation of the joint. But even though the cartilage has been a main focus point, the subchondral bone has been something that people have overlooked, and it's been more getting traction over these past 2 decades in the research and in the literature. And here, and for people who might not know what this is, the subchondral bone is that bone beneath the cartilage that plays a crucial interface with that has an interface with the cartilage called the osteochondral junction. And with the subchondral bone, it has a big role in playing with shock absorption and just supporting the cartilage. And literature has shown that when you give these rats like a high-fat diet, there's changes with the subchondral bone, such as an increase in the subchondral bone plate thickness, sclerosis in the subchondral bone, and there's lesions in the bone marrow, all negatively impacting the bone. But although this is all interesting at the level of OA, one thing that has always like piqued my interest ever since joining the lab was something that you guys might remember from Mauricio's presentation. At the level of the muscle, there seems to be a change in its integrity. And what I mean by this is that when we compare type 1 and type 2 fibers being that's low oxidative potential fibers, they typically have increased amount of fibrosis and fat infiltration, whereas type 1 fibers may be something seen in the soleus muscle that has these high oxidative potential is protected against this fat infiltration and fibrosis stimulated by a high fat sucrose diet. And this connection kind of leads to this idea that there might be some type of oxidative potential and metabolic health when we look at something such as aerobic exercise, because aerobic activity increases the amount of oxygen and activates oxidative pathways, which enhances metabolic efficiency across tissues such as the bone, such as the muscle and the bone. So this is why we want to look at aerobic exercise. If we look at some previous work such as Anada's again, we're able to see that when we look at these joints in the knee, when we leave these rats with a sedimentary lifestyle and with a high-fat, high-sucrose diet, their joints are compromised. Here we can see that the cartilage has collapsed, showing this also this compromised joint. But when we look at it from an exercise plus a high-fat sucrose diet, the joint looks healthy as I showed you guys before. And then when we look at this from a scoring system, we can see that this high-fat, high-sucrose diet with this sedimentary lifestyle has an increase in this OA scoring system. This shows us that aerobic exercise protects against metabolic OA. And then when we think about these two things, one being the muscles, that they have a difference in their oxidative potential against a high-fat sucrose diet, and we're seeing this exercise being an important factor, this kind of leads us to this idea of aerobic capacity. And aerobic capacity is essentially the maximum amount of oxygen uptake your body can use during intense exercise. And if you may know, it is polygenic in trait, both genetic and environmental. That maybe in early life, it has maybe a genetic component, but as you progress over your life, the environmental factors start to play a role, and these two areas comprise of your aerobic capacity. And interestingly enough, this presentation is centered on the genetic component of it. And maybe many people are like, how do you achieve this genetic component? Well, one thing that our lab is looking at is in achieving this inherent aerobic capacity is using this selective breeding model. And in 2001, Lauren Kosh-Shu in the University of Toledo would collaborate with the NIH to develop these two strains of rats. These high-capacity runner rats, like the Usain Bolt of rats, and then these low-capacity runner rats are like the lazy cousin of Ratatouille, if that makes it easy for you to remember, are able to like selectively breed these two and get the two extremes of aerobic capacity. And basically here we could see that over a multiple of generations, we're able to get to this 49th generation. And by the 49th generation, we're able to have this 1,200% difference in running performance between these high and low aerobic capacity rats. And these rats are quite interesting because they've been used in various different studies. One of the studies that have been found when you feed these rats with a high-fat, high-sucrose diet is that it seems to have increased insulin sensitivity, fighting effects of the negative effects associated with diabetes, and lower systemic inflammation. Quite interesting when we're looking at this high-fat, high-sucrose diet. But when we just look at the general effects without this high-fat diet, we're able to see that there's a longer median life expectancy with these high-capacity runner rats and reduced myocardial fibrosis. But although this is quite interesting, the main premise, as I stated earlier, was related to OA. And none of these rats have been actually used to look at the progression of OA. And this leads us to this idea that aerobic capacity might not only protect against metabolic diseases, but also may have a potential role in preserving joint health and degenerative condition changes in the tissues associated with metabolic OA. So to do this, the main objective of this study was to see how inherent aerobic capacity influences or interacts with this high-fat, high-sucrose diet to see if there's any changes across the structural integrity of the shoulder joint. And with this, our hypothesis is that a high aerobic capacity will protect the shoulder joint against subchondral bone degeneration and cartilage degeneration in the presence of this high-fat sucrose diet. So before getting to results, we need to understand the methodology that was approached with this study. So to do this, we were able to get these 36 and NIH rats, 36, all of them being male, and we were able to divide them based off their strain and this diet. This burger and donut is this high-fat diet, and these little mini cubes you see here is a standard chow diet. We were able to feed them at starting at 25 weeks for over 12 weeks and terminate and euthanize them at 37 weeks old. So to see if this high-fat diet worked, we need to see changes in the body fat percentage. So when we were able to get this, we were able to see that with these high-fat, high-sucrose diets, they had an increase in body fat at a rapid rate. Whereas this chow diet also had an increase, but more so at like a slow, steady rate. And after 12 weeks, when we looked at these rats, it's like as we thought it would be, the high-fat, high-sucrose diet had a higher body fat percentage than these chow diet rats in both the aerobic strains. And this is also important, but one thing that we also need to notice is that I said to you that this is metabolic OA. There must be some type of disruption in this metabolic component of it. So to look at this, we got cytokine measurement from the blood. And at baseline, we saw no differences across all the groups. But when we look at these results after this diet intervention, we see that these three pro-inflammatory molecules, MCP1, interleukin-1 beta, and GM-CSF, we see that these low aerobic capacity rats generally have a higher pro-inflammatory marker in comparison to these high capacity runner rats when we look at this high-fat, high-sucrose diet. And then maybe here on the GM-CSF, we see this chow diet having a higher value than the high capacity runner rats when we look at the dietary interventions. And we can see that there was metabolic disturbances in these rats, and that these high capacity runner rats that a high-fat, high-sucrose diet express lower pro-inflammatory markers than these low capacity runner rats. This gives us the idea that we're seeing metabolic disruption within these rats. Now with this project, there are two primary outcomes that I want to look at. First is the subchondral bone degeneration and the cartilage degeneration. So to do this, we were able to put these joints into a micro CT machine, and we were able to get 2D images as it rotated around the machine and get a 3D overall visualization of these joints. And we were able to get sections and contour these joints to get the overall structure of what is happening at the subchondral bone. Here, we were also able to see changes with to do histology. We were also able to get these joints put them under NDF and decalcify them, and put them under paraffin wax to put them under this microtome setup right here in the middle, and cut throughout the joint. And this allows us to get these images and stain them and get this like nice colorful image of what the cartilage looks like. And to remind you guys, as I said for the subchondral bone, one of the few parameters we wanted to look at were these here on the right. Bone mineral density, changes in the bone, like such as the surface and volume, and changes in the trabeculae. Here's what 3D image of the bone looks like. And on the right is what this looks like when we take the sections. This on the bottom is what I believe the humerus, and on the top is the humerus, and the bottom is the scapula. And from here, we're able to see that when we look at these degeneration results, that with these low capacity runner rats fed a chow diet, we're able to see that there is a development of these cysts. But it is important to note that this wasn't the main degeneration we saw. We saw the development only in maybe 2 to 3 of these rats. And the main differences that we saw with these rats were these frayings that we saw. And the fraying right here is like almost like the splitting of this cartilage. And because of this, we're able to see that there were only mild changes observed with these rats. But when we look at these high capacity runner rats, we're able to see that there was no changes at all. Sorry for this change in the image right here, but here on the top is the scapula, and on the bottom is the humerus. And we're able to see that there's usually either really nice clean images where there's no degeneration at all, or maybe just very slightly minor. But we just can't compare images on their own, so we wanted to get a scoring system. And to do this, we were able to do through a modified Mencken scoring system, where a higher score means more degeneration, and a low score means no degeneration at all. And when we get these results, we're able to see that there is no differences across all these animals. But as you can see here, there are 2 outliers here when we look at these high capacity runner rats. And when we did the statistics on these, we're able to see that these 2 values are 3 standard deviations above the mean. So when we remove these outliers, we see a difference between the high and low capacity runner rats fed a chow diet. But interestingly enough, that when we look at the level of the subchondral bone, we see differences with the bone mineral density. It seems like we see differences both in the diet and strain. When we look at bone volume, there's only a difference between the aerobic strains of these rats. And when we look at bone volume fraction, it is similar to what we see in the bone mineral density, both a strain and dietary intervention. What is quite interesting about these results is that when we look at these low capacity runner rats, if we look at these graphs, they typically have higher scores than these high capacity runner rats. It seems like maybe body weight is playing a factor in why we're seeing these increased results. And these could contribute to this mechanical loading, causing this bone remodeling we see here. But when we look at these, for example, when we look at the bone mineral density, it seems like this high fat, high sucrose diet has lower values than the chow diet when we look at bone mineral density. This could be because of those elevated cytokines we saw. And this could maybe play a role in why we see a decrease in bone mineral density with these rats. But when we take these results and look at the level of the trabeculae, we see that here with the trabecular thickness, here as a picture to see what this is, the thickness between the 2 trabeculae, that we see that these low capacity runner rats have a higher value than these high capacity runner rats, similar to what we saw with the bone mineral density and bone volume fraction. But when we look at trabecular separation, the separation between the trabeculae, we saw no differences across all the groups. And when we take this also a step further, we also see no differences in the trabeculae number and the number of trabeculae that we see within these joints. The trabeculae thickness could also maybe be implied on what we saw with the bone mineral density and bone volume fraction, that maybe these low capacity runner rats, because of their loading and higher body weight, may be contributing to these results we see, and maybe potentially the differences in the cytokine level. With these results in mind, we're able to see that there are changes in the subchondral, that the changes in the subchondral bone were more pronounced than we would have seen in the cartilage histology. And it's important to recognize that the subchondral bone both plays early and late stage OA. By seeing these changes in the degeneration of the cartilage, maybe we're playing an early stage of osteoarthritis. And that since the both diet and aerobic strain are playing an influence, maybe having like a longer dietary intervention would be very useful. And when we look at this from previous results, the knee joint seems to be more affected than the shoulder joint. And maybe with these rats specifically, with their shoulder joints, they seem to be more protected than what we saw in the shoulder joints in other animals. This goes back to our hypothesis that maybe that high inherent aerobic capacity provides a protective mechanism against the negative effects of a high-fat, high-sucrose diet. But with this study, there were a few limitations that we were able to see. That it was a preclinical model that we were using animals to understand the basic science of these metabolic OA. And the shoulder joint isn't entirely a non-weight bearing joint. It is a low weight bearing joint that might have an effect on how we see these results. As in humans, it's not at the same extent with. Future directions should be to assess this metabolic OA in the shoulder through this aerobic exercise intervention with these high and low aerobic capacity rats. To do biomechanical testing on the subchondral bone, maybe to see if there's any differences, such as changes in the elastic modulus, or any of those bone parameters that we might see. And to see if there's any sex-dependent differences, because even in some of the previous studies, there's been differences between what we see in male and female rats. And I would like to acknowledge everyone here. I would like to acknowledge two people specifically, Darsh, who really helped me understand how to use the micro CT, and answering my 11 p.m. calls, answering all the questions I had. And Ruth, who taught me how to do histology and why it's important to follow a specific methodology in completing research. And I would like, these are my references, and thank you for listening to me.
Abel, talk to me a little bit about the pathway of shoulder OA in humans. I'm assuming that the reason humans would develop shoulder OA is because they have sort of a history of shoulder injury.
That is one of the more common ways individuals develop shoulder OA. Typically, they might have something, like maybe if you were like, I don't know, like, for example, if you were like bench pressing something or like lifting something, and if you over like extended it, it might have caused like some compromisation of it, or maybe if you like even dislocated your shoulder, that might potentially lead to that in the future. But there are also some individuals who just develop it like without any injury, and that could also be a player role, but it's been shown that injuries are like a big factor.
Yeah, so what do you think would happen if you tried to introduce some kind of instability to the joint in your animal model?
Actually, that would be quite interesting because previous models have also looked at, let's say, the knee, and if you got like an ACL tear, so they would like introduce like some type of tear in the meniscus in that area, and they did see development of severe OA in those knee joints. So I wouldn't be surprised in the shoulder joint if they did some type of like an intervention with the shoulder that we would see also OA in that area.
Yeah, it might be interesting to see how the high capacity, low capacity rats would differ in their response if you introduce some kind of, injury to the model. Thanks, Gordon.
I also had some questions about the shoulder, because in these rats, of course, they're quadrupeds, so how, can you just speak to a little bit about how that would differ, like, for humans versus what you see in the rats?
Well, with the main quadrupeds, they're usually more time either on their forelimbs, or sometimes they might be on the occasion, maybe if they're trying to drink something or eat something, they would go on their hind limbs. But most of the time, they're using all their forelimbs, whereas for humans, we're not always putting like weight bearing joints on, like something weight bearing on our shoulder.
Right.
We're just usually just walking, but there is a gravity playing a factor with it. There's also like I guess like also the forces with the muscle, like maintaining that stability. And then other things like that, so maybe it's not as, the changes won't be as pronounced as we see in these rats. But there is that still weight bearing effect, because it's not just purely non-weight bearing. There's still like factors, like joint forces that are playing a role.
Thank you.
Questions for you? Go ahead.
I don't understand those blood markers that you were showing earlier. It seemed to me like the high capacity, high fat diet animals had lower pro-inflammatory markers.
The low capacity?
the HCR, high capacity. right?
Yes,
And then they have the bad diet.
yes,
They have the lowest pro-inflammatory marker. So lower pro, I have to figure out what that means. Lower so pro-inflammatory is bad, right?
yes,
So lower is better. So why isn't the,
like, why isn't the
why
diet
does the
causing it?
bad diet give you, a lower pro-inflammatory response?
That was that is a good question. Even I was quite surprised when we got these values themselves. To be honest with you, I don't know entirely the entire reason why it could be that. Maybe that there's maybe some type of change with these high capacity runouts, maybe even like maybe potentially like the diet isn't really doing it, the entire change, maybe the 12 weeks. But like, I do have the same idea as you. I don't know why they're not changing. Our lab thought the C cytokine levels would be higher in the high fat sucrose diet, and the chow would be like lower. But yeah, no, that is fine. Maybe there's like this strain effect that's maybe playing a role with these rats.
Does the diet do anything to how much they're active? Like having the high fat, high sucrose diet, does that make them like super active? Which is not what I would expect, but.
No, like what we expected, like with the cages, like these, like the ones that are high fat sucrose diet, like they tend to get more weight, and they're like, I guess they don't move that much, so they stay more sedentary. Yeah.
Just, I found that.
Okay, and then I have another question about the trabecular bone analysis. These measurements, the trabecular thickness, trabecular spacing, trabecular number. Should, if sort of the region of interest is fixed, they should be relatively dependent on one another.
Yes.
So if trabecular thickness goes down and number stays the same, you would think that spacing would have to go up. So what is happening here?
That could potentially lead into like the voxel size we chose.
Okay,
So maybe like going with like the threshold frequency we use for this micro CT, that might have been like changes with how we saw these values, because if we chose like, I believe I'm right, a lower voxel size would increase like the accuracy, or more so we would see more of the quality of the bones instead, but that does lower the region of interest because of that. And I believe that is one of the main factors we saw with these joints. Because in the knee joint, we did see changes with the thickness and separation, I believe.
okay, and if the number stays the same and the thickness goes down, you should have a lower bone volume fraction. So you have a lower bone volume fraction for the high capacity, right?
Yes.
Yeah, okay. So that makes sense. I just wonder. I don't know, I guess it could be a resolution issue that, when you conduct that trabecular analysis, you pick the exact same region between each rat.
Yes.
And everything. Okay,
That was like one thing we wanted to keep consistent.
yeah.
Corey, you have another question, and then.
Yeah, I was just curious, do you know, like, if there's differences in the development of OA depending on what type of joint it is, like how it articulates, like a hinge joint versus the ball and socket joint?
Not, like more so like, for example, like a hip joint.
Yeah,
Like for these rats, one thing I would like to state is like when we're looking at it from, let's say knee, shoulder, hip. It seems like the knee is the most affected, then the shoulder, then it's the hip joint. So potentially with some of these joints, you might see lower amounts of degeneration or changes across your joints. So for example, if we were to look at like, let's say the hip joint, we would see almost historically speaking in our lab and like other researchers, the hip joint, we wouldn't see that much differences. I don't know why, but like overall, those ones are the least, but the knee joint has the highest.
do you have any like
Problem.
speculation as to why?
One of the things is maybe the fact that the shoulder joint doesn't have like that interpatellar or intrapatellar fat pad. That's been noticed that has a role in like the amount of local inflammation. So that could be a factor. Like that was a big factor in some other papers that they have acknowledged. And also, I believe it's also the idea that there's more muscle like attachments point to the shoulder joint, whereas the knee doesn't. So maybe that could also be a factor for them.
Thank you.
Daniel, you had a question?
You mentioned something about the function of cartilage. You repeat that.
Oh, yeah, no, the cartilage between the joints allows, it's like that smooth layer we have. That basically it just allows us to have like that nice easy movement we have every day. Whereas individuals with OA, it's quite difficult, it's like stiff and painful. And maybe even some individuals notice like they hear like crackling sounds with the joints. So that just overall just
What is the function of cartilage?
hard.
What is the function of cartilage?
What is the function of the cartilage?
Oh, just to help us have that lubricating, that smooth movement that these for the joints.
You said something about damping.
Them being
You said something about damping.
Damping? Did you say something about the damping effect of cartilage?
damaging. More, I think that what I meant in saying, like over time, like if you have OA, there's like that damage you have where that cartilage goes away, then you have those OA symptoms. But no, I don't know about the word, dampening.
Okay. Then you have that picture at the beginning with two gentlemen with the high forces of the knee and the high forces of the hip.
Here?
Yeah. And you mentioned something like that because they are obese, they have higher forces in the knee.
Oh, yeah, like, the excess body weight adds like more forces on like the hips and the knees. And that could potentially be like that wear and tear we see with like OA degeneration. Like that's like the traditional train of thought for OA. That because of that excess body weight adds more forces on
Which
the joint.
suggests that OA is dependent on the forces.
It does have effect, yeah, forces do have an effect on the for OA.
Are you sure about that, or are you speculating?
Pardon?
Are you sure about that?
Like that's body weight?
Yeah.
From what
I think he's asking is, are you sure that OA,
That the forces
that the
have.
forces have anything to do with the development of OA?
Are you sure of that or are you speculating on it?
more so, from what I've heard, like more so like I guess speculating, but like a lot of people have been like making that claim. But a lot of people have been trying to go against that claim that it's entirely based off that. But there is a contributing factor with that.
That have not been shown.
Pardon? Like definitively? From my knowledge, I don't know if I would say definitively for that. But it does play a factor because also you can also remember like it also with that adipose tissues even playing like the forces on them. Other factors like the adipokines and cytokines can also play a role with that degeneration. So I wouldn't say 100% of it is because of those joint forces, but the weight potentially has an effect on it.
Other questions for you? We have online.
Okay, go ahead, Walter.
All right, Abel, can you hear me?
Yes.
Okay, yeah. I was wondering because, I remember a while back you wrote the ORC abstracts and you had some results. And today you show more results. And I remember the result that I found was so interesting in your abstract for ORC was that you had differences in the bone between the two phenotypes, between the high capacity runners and the low capacity runners. And then you also had differences in the bones as a function of the diet, but you only had that in the low capacity runners and not in the high capacity runners. And today the results went a little bit too fast for me, the additional results that you showed. And I was wondering, is that still correct? That the diet has no effect in the high capacity runner, but has an effect for some parameters in the low capacity runners?
For the bone results?
Yes, the bone results, yeah.
So like the ones you see on the screen?
High capacity, it's not significant, not significant, not significant. But there is
Yes.
differences between the phenotypes, yeah, and you showed more results. What were the other ones? Here as well, yeah. So again, no differences, no differences, no differences in the height. Yeah, so it seems to hold up what you had for the couple of results that you showed at URC, that
Yeah.
there seem to be distinct differences in the bones between the phenotypes, the low capacity and the high capacity runner. So first question, how do you interpret that result, or what does that mean to you?
Oh, that like potentially like an inherent aerobic capacity, like if one is genetically like determined to have a low one, they would maybe have more, that their bones would kind of like change based off that. But with also like with the diet, that also can have like a stimulating effect on how much change we see. So here, like let's say like if we look at the thickness and separation, it seems like the strains have more of a potent effect on them, seeing that difference. But maybe the diet itself isn't too much, or maybe playing like a small, has like a gradual role in making those changes.
I mean, I'm a very simple person. To me, what that result suggests that inherently the high capacity runners and the low capacity runners have different bones, and it's shown as a statistical difference in some aspects of bone parameters that you measure. So they do have different bones. And then the second very simplistic interpretation that I have is that for whatever reason, the bone that the low capacity runners have seems to be sensitive and seems to adapt and seems to change as a function of the diet, whereas the high capacity runners, they seem to have this stable bone that doesn't seem to be affected. They can eat a chow diet or a high fat sucrose diet, and it's not affected. And would you agree with a more simplistic interpretation like that?
Yeah, that makes sense.
Because to me that's a fabulous result, and I thought if I would have been you, I think I would have emphasized that a bit, that with the high capacity runners, you seem to be able to do whatever you want in terms of diet, and it doesn't seem to affect the bone, whereas in the low capacity runner it does. And not only that, there seem to be lots of differences between the high capacity, low capacity runners. So they seem to have two different bones, and one bone adapts more to the diet and the other one doesn't seem. I think that's a really neat result, actually. Just as a comment, I guess no answer required. Thanks, Abel. That's all I wanted to say.
I had a question, Abel. Give some explanation on why the high capacity runner rats had lower bone mineral density?
Yes.
I didn't catch that. What was the reason for that?
Oh, it's because of the potential of like maybe the different kinds of cytokines that kind of like stop that like progression of the development of like bone mineral density. Those have been like negatively affected. So that can maybe like reduce the amount of bone, like the quality of bones.
So the high capacity rats have more of that?
No, well, if we're just looking at, because this is also just in like normalized or like their body weight, for example, so like these low capacity runners have higher amounts over in comparison to this high capacity runner. But when we just look at the diet effects on each on their own, like say these low capacity ones, it seems like the diet has more an effect as like what Walter says on their bone, but that negative effect of those like cytokines, for example, negatively impact the amount of bone quality it can have in comparison to the.
Any other questions for Abel? Yes?
And I don't think you did any mechanical testing on the bone.
No,
So do you have any speculation? Like, do you think it's
potentially because we're.
vaginal?
I wouldn't be surprised, maybe they could be, but it is quite interesting that, for example, like this, like these values aren't like normalized with body weight. Maybe that could be playing a factor in how we see these results, but we could see the low capacity runner rats having more fragile bone in comparison to these high capacity runner rats. And as maybe in this, these changes could be sent maybe from, if we do like cyclic loading tests to failure, maybe we see this more damage with these bones of these low capacity runner rats.
So do you think it's a chunk before then? So do you think this is a property of the bone in the animals? Because this is subchondral bone. So do you think it represents the bone?
No, I don't believe the entirety of the bone, because there's also this bone, like for example, if we look, I believe we looked at the humerus proximally, we didn't look at the distal aspects of it, so that may be like a whole joint thing, like a whole bone thing. But just at this level, we're just looking at like a small portion of these like shoulder joints, like the subchondral bone.
Another online question?
Yes, go ahead, Walter.
Yeah, good. Yeah, Abel, I was wondering, you have a lot of inflammatory markers from your blood results, and then you have a lot of bone results, these various parameters that you measure in the bone. And I was wondering, have you tried to run statistics to see whether or not some of these, because the variations in some of these inflammatory markers are quite big. Have you tried to correlate these inflammatory measures with your bone measures and see if there might be some relationships between one or the other?
I have not. That would be good to look at, but one thing I kind of did notice with these data is like, those two outliers. One of my ideas that I had was maybe potentially like the cytokine levels could be attributed to those like higher damage in those two high capacity runner rats. But it seems like those two, the cytokine levels weren't that high for them. They were actually maybe even like average or like maybe slightly below. So maybe, I don't believe, maybe we'll see some kind of like relationship between these pro-inflammatory molecules with the changes in the bone, but I could look into it to see if there was maybe some kind of like moderate correlation with it.
Yeah, I think that might be neat to just, include it or exclude it and see if there potentially might be a relationship. I remember Kelsey Collins did that, and it was quite remarkable, between the gut microbiota parameters she found, and also between some of the inflammatory markers, she found quite high relationships with the Mankin scores. And so here, and also what could be done is maybe take the body fat percentage of the animals and relate that to the bone parameters, or also relate that to the Mankin score, or also relate that to some of the cytokine scores. So in order to get a bit more insight of what might be happening mechanistically, run these statistics and just see what happens, even if most of them are excluded and don't give anything, but just including or excluding them. I think that would be important to do, just to kind of finish up the analysis.
Okay, I'll look into it. Thank you.
Thanks.
Right.
Yeah.
Well, one question there and then Brent.
So other than the blood, cytokine, do you have the local cytokine?
Pardon?
So the cytokine measurement inside the joint.
Oh, no, these cytokines are like from the blood, so they're like giving us that systemic changes. I didn't get local inflammation markers for the rats, not yet, because the synovial fluid is quite low within them. So we couldn't, I wasn't able to collect local inflammation markers for these rats on the shoulder joint.
Okay, so I think I figured out why you could have a decrease in thickness with not an increase in spacing. If your total volume decreases, and I think that's happening. And so if you go to bone volume fractionaire, you see how the bone volume is lower for the high capacity runners, or
Yes.
whatever they are. And then that difference kind of gets exacerbated a little bit when you go to HFHS, meaning that the total volume is less for the high fat, high sucrose animals. Now the question is, why do animals on the high fat, high sucrose diet have less total volume?
Oh, like of the bone?
Yeah, like it's bone volume divided by total volume. And I don't really know how you conduct your analysis. I don't know if you like pick a cubic region or if you segment out, like, do these animals have smaller bones?
From a visualization standpoint, I did not look at the size of the bones because I didn't want to say any potential biases. But to be honest, why the high fat sucrose has a lower bone volume?
No, I'm
Sure.
telling you, well, I'm saying, and then I have a question. So the high fat, high sucrose animals clearly have less total volume that's being measured. Right, so the way you get BVTV is you have a volume.
Yeah.
That you're analyzing, that's your total volume. Some voxels are bone, some are not.
Yes.
So for some reason, your high fat, high sucrose animals have less volume
Both, or the,
No,
the
just
total bone.
everything being analyzed is less. You're analyzing less bone. So how are you doing the bone segmentation? What location is the bone being analyzed in? Is this just the subchondral bone?
That's the subchondral bone.
And so do you say, okay, like this is where the subchondral bone starts
Yes.
and this is where it ends?
Yes, we did,
So then that means that would say to me that the subchondral bone thickness, which is a different measure that maybe you should be looking at. Subchondral bone thickness in the high fat, high sucrose diet animals is smaller. They have thinner subchondral bone. And if they do, if you quantify that thickness and it's thinner, then I think you can explain anything. And if they do have thinner subchondral bone, then that's very interesting, right? Because that is something that's believed to play an important role in
yeah,
the development of
yeah, the plate thickness.
the plate thickness, yeah.
Yeah.
Normalize to something, then to the length of the bone, to the size of the animal.
No, this is great. I'm so used to analyzing sort of square regions of trabecular bone where the total volume doesn't change them. Or, you segment out sort of a whole metathesis or diathesis, so. But if this is just the subchondral bone, then no, I think it's right. It's just that I think you need to kind of understand and report everything and figure out what's going on. But I think it's important that the subchondral bone plate is thinning when you have the
High fat.
high sucrose diet.
Alright, if there are no more questions for Abel, let's thank him for his talk.